Silphium Prairie at we'll be hearing today about the summer floristic survey of Silphium Prairie at Rock Creek Ranch Park. This is part of the Armchair Botanist Forum that we have once a month, and I see a lot of familiar faces and perhaps some new faces that are here in the participant list today, so welcome to everybody. The Armchair Botanist program is a collaboration among botanists, museum professionals, students, biodiversity informaticians, and all of you. These casual forums that we hold monthly will serve as a space for questions and discussions with current digitization projects that we have going on, and a chance and opportunity for both presenters and attendees to interchange information and share information about plants. So thank you all for being here. Please remember to be respectful in your conversations. When we do start the official presentation, please go ahead and type your questions directly into the chat, and we will answer them at the end of the student presentation today. If you have any questions or comments that aren't addressed here, we'd love to hear them. So please send us an email at herbarium@brit.org. I will throw that email address into the chat here. All right, and I'm reading the chat right now, everyone. Welcome, Sue from Waxahachie, and I see a lot of other folks in here that are familiar faces. Let's go to the next screen. So those of you that may not be familiar with the Armchair Botany program, this collaboration is an effort to digitize the scientific holdings that we have in our herbarium here in Fort Worth, Texas. It's part of a much larger global initiative to digitize these important natural history specimens that can inform us about our natural world. And what you're looking at on the screen right now is the general process of how this happens, at least here in our herbarium. And you'll see there's a clear point in there in which armchair botanists, scientists are helping us extract data from specimens. Today, you will see a little bit of all of this from the very beginning. So without further ado, it is my absolute pleasure to introduce the 4 young people that will be talking to you today about their summer project. Up on this screen, you're seeing from left to right, and not necessarily in this presentation order, we've got Jade Affleck, Cade Morton, Gabrielle Perez, and Gabriella Wolf. These 4 young people were supported over this last summer in this internship through private donations to our organization. And they have collaborated, you'll hear a lot more about the project, but they've collaborated with the city for a really great example of what we can do here locally. So without further ado now, I will hand it off to our conservation and collections interns from this summer.
Okay, I'm going to share my screen. Can everyone see that? Perfect. Okay, so I'm Jade again. We're really excited to talk to you about what we've been working on this summer. So I'm going to go ahead and get right into it. So Rock Creek Ranch Park, or RCRP, is a 303-acre reserve park, and it's owned by the city of Fort Worth. It's located in Tarrant County in the Cross Timbers ecoregion, and the city of Fort Worth is the 4th owner to own it. It was previously owned by Alan Meeker, and 275.5 acres came from him, and then the other 27.5 acres came from the estate of John Lewis Merrill Sr., who is pictured to the right. And he is sort of this icon in the field of range management and land stewardship ethics. He's been ranching the land for like over 40 years, and he also built the house that is on the property. So historically, the park has been used as ranching land, and it also contains an old fracking pond on the property. The master plan for the park is set to begin in 2026, and there's expected urbanization to occur near park boundaries. Also, the Tarleton campus in Fort Worth is very close to the park, and they're expanding their campus. Some of the pictures to the right, that is just a creek that runs in the property, and then there's also, this is a stone wall remnant that's from the original homestead, so that's pretty cool. So our work was focused primarily in Silphium Prairie, which is just this small hatchet-shaped area. We have a map later on in the presentation that will kind of show you the parameters, but it contains high-quality native prairie with several endemic species. Its geology is composed of early Cretaceous period clay and limestone, which gives rise to habitats such as seeps, barrens, and as we were walking around and observing, we noticed these microhabitats, kind of like little divots down where it was also moist and different species located there. And these are all unique to Fort Worth prairies. So these are just some of the iNat observations that we took of the endemic and species of interest. And what I mean by species of interest and these indicator species that I'm about to talk about, these plants are characteristic of Fort Worth prairies, and basically their presence indicates diversity and nativity at the park. So starting on the left with the Silphium, this is what the prairie is named after, of course. And then the seep muley, we saw the spike rush, little blue stem, this is Texas prairie parsley. And then some other indicator species that the city had listed that they're interested in that we observed but are not pictured are palm leaf, wine cups, the antelope horns, and then prairie preetleaf and Drummond's skullcap. And we all have these in our iNaturalist project that you'll see later. So we are, like Tiana said earlier, the conservation and collection interns, and kind of our objectives for this summer and for this project was to provide the City of Fort Worth Parks and Rec Department with information about the prairie so that they can best create their master plan. Not only, we were also vouchering and digitizing plant specimens at Silphium Prairie so that these holdings can be accessible to the city and anybody else doing research out there. And then we also got to learn lots of new skills at our time here. Not only were we vouchering plants, but we got to do seed collecting actually last week, and we did the Silphium because it was only right. And we got to learn the ethics of conservation and collecting. We got to digitize and image our own specimens, which was really cool. We kind of got to go through the whole process of taking a collection and depositing it into an herbarium, like from the field to depositing it. We had to create the labels for them, which was such a process, and we'll kind of talk about later, but we all figured it out and kind of made our own unique labels. Tiana showed us around Symbiota and Torch, which Kate mostly worked in. She made the checklist for us, so she'll talk about that a little bit later. We did make our map in QGIS, which Gabby made the map, so she'll show you a little bit what she learned. And then we got so much experience in plant identification. We all kind of came into this internship not really, we were out in the field and not clueless, but we were not able to like kind of point at stuff and know what they were yet. And, kind of towards the end, we were able to go out there and know what stuff is. And then having to key out all of our own specimens has really helped us be able to know what characteristics we're looking for and how to identify these plants. And then we also worked heavily in iNaturalist. So all of our observations and what the city will be kind of using to look at what's out there is going to be in iNat. So all of our like pictures and observations are there as well.
Hello, can everybody see me? All good? Cool. So my name is Gabby Wolf. Not the Gabby that did all this stuff with the QGIS, but there's two of us. Anyways, I'm getting off topic. So here I really just kind of went through the workflow and the methods with what we're doing. So like Jade was talking about earlier, we really did go through the entire process. From start to finish, or at least almost the entire process. So what our days typically consisted of was at least once a week, we'd get up early, go out into the field, and we would, our method of surveying was kind of more of a meandering walk to make sure that we really get everywhere. And then what we would do is we would find plants with characteristic features that would allow us to figure out what they were whenever we can, get back here and key them out and all that, but that's a little bit later. But what was really important for us to look for was the fruits, the flowers especially. And so we typically pretty much only looked for typically herbaceous, but definitely like the fruit bearing and flower bearing plants. And kind of with the ethics, you got to make sure that there's a lot of that plant species in the area so that you don't overtake from the population and impact it negatively. So we kind of had to make sure that there was around at least 10 in the area to feel comfortable taking 1 to 3 of them to make, a duplicate, triplicate, all that, which is great for scientific study. One other thing that's really important is whenever we go around and, dig them up, if possible, getting the root is really important because it is kind of diagnostic of whether it's annual or perennial, or there might be some really interesting morphological characteristics, like if it's a bulb or something like that. Additionally, as we were walking around, even if we weren't collecting that day, because we typically were in pairs and one person would do a majority of the collecting while the other person kind of helped them, we would be posting on iNaturalist. We would either take photos and post them all in the field, or we would use iNaturalist as a tool to help us field identify, since, not all of us had great field identification skills of the native plants here, especially me. It was a really, really great tool to use to kind of see what we're getting so we don't collect the same thing over and over again. Now, whenever we get back from a field day, or actually, let me back up a bit. I realize I missed a little step. So in the field, we would also quickly just press the plants into a field press. It was pretty much cardboard and then some newspaper and some more cardboard. And that was a great way to kind of temporarily store our collection. We would write it with our collection number. And then at the end of the field day, we would come back to Britt. And here we are in the process voucher section. You can actually see in the photo, we are repressing our specimens. Here we used cardboard. We still have that, oh golly, I forgot the word. What's the word? Newspaper. We would use the newspaper and then we would add blotting paper on top of it, or underneath it. It was a little kind of just how wet the plant is. And then if it was a really bulky plant, you would put foam on it. And then you would just sandwich it with another piece of cardboard. And we would do that with all of our collections for the day. And then we would put it in a wooden press, which is a lot more sturdy than a field press. And we would put that into the large dryer. We'd sign it in. And after probably about, what would we say, like 2 days? About 2 days, they were typically good and dry, unless you were like me and like to get a cactus, which took probably about a week to completely dry. Then after that, to continue processing the voucher, we would go and after they are dry completely, we would place them in a freezer. And after about, say, the weekend or a couple days, we would get rid of all those pests and stuff that we really just don't want eating our vouchers. After that, they were carried back typically to right where we are in the herbarium to be keyed out by us. So we used the dichotomous keys from the floor of North Central Texas to go through. We would typically use the iNaturalist genus and species that they gave us to kind of check ourselves. And we would start off with the family, making sure that we had all of the correct characteristics, move down and continue going through piece by piece to see what species we had. The photo below shows, I believe it's pronounced a croton, and that the genus croton. They have very, very interesting flowers. So I really wanted to get a closer look through the microscope, which has been super cool. But I loved that photo, so I wanted to throw it in. But another diagnostic characteristic that you often look out for is what are called the trichomes. It's those little crosshatchy looking hairs right there. At least in this species, it's crosshatch looking or star looking. I think stellate is the right word for it. But then after we finish keying out our specimen, we would record it in our collections log along with the species author and all that sort of stuff, getting that filled in so that we can refer to it later. When we make the labels, and I know Jade hinted that it was definitely a process. It was using mail merge. We would take all of our specimens, all of our keyed out specimens, and we would put them into the mail merge, and we would make a label. So here, I'm so sorry if this doesn't come out looking perfect, but here is a collection that we made. This is Scutellaria drummondii. And down in the corner, you also see that's one of the basket flowers. And here is the label that we made. Now, I know that I didn't give y'all a chance to read that, but there's a lot of really important information on there. You have your GPS data. You have what you noticed in the field, whether it had a smell or kind of what species were associated with it. You also had its frequency. Was it really common in the region, or was it the only one you saw? And this will tell future researchers kind of what was going on at the time. It also has whenever we keyed it out, whenever we collected it, so it shows the time of year. So it was a really, really important part of processing the specimens and getting them into the herbarium. So now to finally enter them into the herbarium. So with help from our amazing, amazing volunteers, they typically mount our specimens just using archival paper. And archival paper tends to be acid-free. And I think it's cotton paper that we're using. It's stamped with the Brit logo, and they put the specimen on the sheet with some clear drying glue, and same with the label. And after that, it is officially getting into the system. We then put the barcode. I can show it here. We put the barcode here on the bottom, and that will help you identify, what what specimen it is. It helps whenever you go into imaging. That helps you read, or it helps go into the system as reading that same plant and all that. So yeah, I think that was all. So thank you. And then here is the next slide.
Okay, so these are some of the results from our survey, which was specifically from June 5th to August 3rd. So just about 2 months of surveying. In the graph to the right, you can see that I've pulled out the top 8 families that had, 5 or more species, different species observed to it. So we observed in total 51 families and 173 species. And of that 173, we vouchered 105. And you can see that Poaceae was the top family with 31 species, and then the Asteraceae with 30, and then Fabaceae with 10. And there was 43 families here not shown, but they all had less than 5 species observed, and 26 of those only had 1. So there's not much evenness shown, but that is to be expected because the grass and the aster families are the 2 most common in the area. We did kind of see what we expected. And then in total for our project, there was 797 iNat observations. This includes observations and also species that were vouchered.
Here is our Texas native and non-native plants. We actually had a pretty good amount of purely native plants. However, we did have a small percentage of non-native and a bit smaller percentage of invasive. So some of the invasive grasses are like the yellow blue stem, for example, or the privet. You can kind of see which ones are invasive with the little no sign that I put up in the corner. But generally, most of our plants that we found were native to the environment, which is a really good sign. And, some of the grasses were used when the area was used as ranch land. It was used for making hay. And so it's expected to see some of these invasive grasses there, but not necessarily wanted. So yeah.
Hi, I'm other Gabby, and I worked obviously with collections and conservation, but also primarily the QGIS, GIS systems to create these maps to kind of give a summary of what we did. So a couple of features on the maps, I'll start with the smaller one first. It is just a satellite image of the entire boundary of the park. The orange inside the white square is our coverage over what is Silphium Prairie, on a large scale or whatever. The smaller or the bigger, more close up map has more of our information that we summarize and put on here. It includes the 3 largest families that we found here, like the highest quantity, and the 2 other additional features are the green lines, which are our tracks, our GPS tracks that we were able to put in, and it also has the yellow ellipses, which adds the introduced species areas. These are both transparent features to show like the quantity in that area. So the darker means more, lighter means less. These maps kind of show the coverage of the prairie we surveyed, the locality of the most prominent species that we found, and the areas that include introduced species that we didn't know before. And this is all pertinent information to the city and how they will use this area, as well as it adds to the just general knowledge of the area that we've never had surveyed before due to the fact that it was privately owned before and now city owned. Let's see. I think that's about it with what I had. Yes.
Hi, I'm Kate, and I'm going to talk to you a little bit about why vouchers are important. Vouchers give us an accurate representation of plant populations within a certain geographic area, which is especially helpful for restoration efforts. So, for example, in the future, if some organizations or researchers are interested in restoring a prairie such as the Silphium Prairie where we worked, they can refer to our vouchers and see what sort of native species were growing in the area and decide whether they could maybe reintroduce those down the line. They also are very important for preserving morphological traits. More and more studies are coming out showing that climate change is causing species to change morphologically over time. So having these physical specimens that you can track throughout the decades and study the morphology is very helpful for studying these trends. They also come in very handy for comparison whenever you're trying to determine if you found a new species. And then you can see we have a prairie milkweed there. The one on the right is the one that we collected at RCRP. The one on the left is from the 1980s, also collected in Tarrant County. So at the bottom there, I have a timeline showing the species we collected at RCRP that do not have a voucher in BRIT from Tarrant County since 2010 or earlier. And at the top, there is a chart, and none of these species we found had any record of a Tarrant County voucher in BRIT. There's a few reasons for this, most likely due to bias. For example, Centorium tenuiflorum, it is invasive, and a lot of voucher collection projects focus on native species. It also is very close to Zeltinaria texensis, so there's a chance that there were some identification errors in the field, and that might not reflect how many vouchers are actually in the herbarium for it. Solanum carolinense, on the other hand, is native to Texas, but it is infamous for causing something called crazy cow syndrome, And so that negative connotation with the plant might have discouraged some people from collecting it, especially in Texas. So I also studied some of the phenology trends for our species. On the left, there is a bar chart showing some of the patterns in the blooming periods. The premature and overdue species we found in bloom outside of the predicted blooming period, according to the Ladybird Johnson Wildflower Center, while the early species we found in bloom within the first month of the expected blooming period, and the late species in the last month. As you can see, it somewhat supports the EPA study that was recently done, showing that many species, due to climate change, are blooming earlier, but for some reason, there is a section in the northern Midwest and some of the southern states, including Texas, that are actually showing some species that are blooming later than their usual blooming period. Now, it is important to consider that it is likely most of this data, both by the EPA and the Ladybird Johnson Wildflower Center, are using national data for their blooming period calculations. So since we are focused in Texas, there may be some geographic errors in that. Finally, that brings us to TORCH, or Texas-Oklahoma Regional Consortium of Herbaria. TORCH was very helpful for identifying specimens down to the species level, because it provides species images, characteristics, examples of vouchers, and some interactive maps. The interactive maps were especially helpful because we were able to check if the species we suspected our voucher matched with was collected frequently in our area. And you can see down on the right, that's the thistle that was collected in Tarrant County. I have created a checklist on Symbiota that you can access by scanning with your phone in the bottom left corner. We vouchered a total of 87 species, 100 taxa, and 40 families. So if you're interested in learning more about each species we collected, you can scan that.
Alrighty, and now down to some fun things that we just found throughout the park. So we found there has been a little bit of an error in kind of our research for a moment. So the Escobaria missouriensis var similis, it isn't threatened, but the species is often threatened in certain areas by like fire ants and some other things. So it's a little bit difficult, or is it fire ants, correct? Yeah, threatened by fire ants. So it can be sensitive to stuff like that. But we also found this threatened species called Echinacea astrorubens. It is a coneflower. It was actually intermixed with the not rare species. It was a little bit difficult for us to tell because they were no longer in bloom. So we had one of our kind of experts here go in during one of our field days, and she went through and tagged which ones were rare, which ones were not. So that was really, really, really cool and really interesting to see. And here in the bottom, we have our liatris, and that one has some mildew on it. The poor liatris could not catch a break. It kept getting, we kept finding it with infections, and it even had, another one had a daughter plant, so parasitic, correct? Mm-hmm. Yeah, so it had this big orange parasitic plant on it. So the poor liatris just couldn't catch a break. And up above it, we have one of our I'm so sorry, this is a hard word for me to say, curcubita. It is this huge, huge vine. And it's really weird because it smells like Whataburger, or at least some meaty thing whenever you rub your fingers on it. So that was just interesting and funny.
And in conclusion, Silphium Prairie, we found, is diverse and it contains endemic, rare, and threatened species. This implies that Silphium Prairie has a value and deserves to be protected and well conserved, even with park development and additional development around. We provided information that the city can use to make educated decisions on the future protected and natural areas of the park and the best ways to implement conservation efforts for this specific property. The important areas for the city to consider, we determined, are the Seep, the Barrens, and areas containing threatened species, of which there are many. This survey also contributed phenologically and geographically significant vouchers to Brit and the herbarium collection here. And further surveys should be done in other seasons due to the fact that we were mostly in the summer to give a more accurate representation of what is all present. Some species couldn't be vouchered because it was before or after their blooming time. And then we just want to thank so many people. Thank you to everyone who has supported and helped us, from identifying and processing with us to teaching and sponsoring us, including the brilliant Brit staff, faculty, and volunteers, the City of Fort Worth Parks and Rec Department, and the Nicholas J. Martin Foundation, as well as private donors. And we can't forget the iNaturalist community that really carried us there.
We really did.
So, okay, now I think we're ready for questions.
Or no, we got one slide left. So these were just some other fun things that we found. We found some mammal bones. We found this huge tarantula. There's a cute little buggy that's just staring right at us. But also throughout this internship, we all got closer and really got some new friends out of this. And you can definitely tell by the way our body language changes from that very bottom photo of all of us on our very first day to our very last day out in the field, where we just took a quick picture. And we also loved our Starbucks runs during lunch. But also the center image right there is our iNaturalist project. It will take you there and you can kind of just see what all we've observed, and it'll show some of our vouchers. And yeah, it has been a absolutely great time, and we're all so glad that we have done this and that we can show this all to y'all. So thank you. Any questions?
Thank you all for your wonderful presentation. I was there for all of this, but it is wonderful to see it presented like this. You have done us very proud, and it is really incredible. There have been various questions that have been submitted in the chat, some of which fall under our purview and some beyond. So I'll try to address them in the order in which they arrived, and some of these questions were actually answered in the chat. So one of our first questions was, how many years was the land used versus laid fallow? So some of these data we didn't actually have at the beginning of our project. So this is some data that the city themselves are compiling. So Lulu, hopefully that's in Brook Best Road response in here. Thanks, Lulu. We're unsure exactly the last date of grazing, but we definitely did not see any areas that appeared recently grazed. The second question from Lisa Marler in here is, are there any plans for removal of introduced species by a volunteer group for the park system? And the response to this one that was also responded in chat, because this is property that we have been graciously allowed access to by the city of Fort Worth. The response is that it's likely that the city Parks and Rec staff will want to address the problematic species. I believe there were already plans to address Johnson grass in some areas. We've worked closely with 2 Parks and Recreation Department, City of Fort Worth personnel, who would be the ones to pull out a call for volunteers if it comes to that. So Michelle Villafranca, Jared Hall, and Rick Wilson have been just incredible in assisting us with this work this summer. All right, another question from Lisa, and this is one for y'all interns. Bloom dates at the Lady Bird Wildflower Center. The question is, are these blooming dates by location? So this was in response, I believe, Kate, to the slide that you presented.
They are not based on location, and that is why that's something you need to keep in mind when you're looking at it, that it may not be the most accurate because it doesn't account for any geographic changes of it being within Texas. But it still matches up with a lot of data collected by the EPA showing that I think it was the honeysuckle species is what they looked at in particular. The honeysuckle in Texas was blooming a lot later, while honeysuckle is blooming a lot earlier in a lot of different other locations. So I think it's still really interesting, and I think it still could correlate, but to make a definitive theory, we'd probably need to look more specifically within Texas than nationwide.
Thank you, Kate. That's perfect. And your explanation of the importance of specimens and going back to them. And Gabby, your talk about the dates on the specimen sheets are very important and allude to that. So thank you very much. Let's see. We had a question about signing up with the online transcription. Thank you, Katie. I put a web address in there if you'd like to learn more about the Armchair Botanist program, and also if you'd like to see our upcoming forums. Let's see. A question from Lulu. Have there been or will there be iNat-oriented bioblitzes there to further document more species? And so the answer to that question, Lulu, is yes. There have been bioblitzes out there, at least one that I'm aware of, and there are plans for, excuse me, doing more in the future, but that would be another City of Fort Worth call out for bioblitzes. So hopefully we can get some of these interns back here to help participate in some of those. There's a, oh, well, actually, let me tag on to that question. Interns, if we're considering doing bioblitzes, do y'all have any comments on or any advice to people doing bioblitzes? You have had to identify from images only and from specimens. So any advice to iNat users documenting these prairies?
Yeah. I can do it as said.
Yeah, sure. Yeah, right.
100% please get the flower. I had so much trouble because there was often times where I did not have a good enough photo of the flower. And once you're getting down into the species or variety, it is sometimes imperative that you have it. There was this one, actually I can probably grab it. This bugger right here. It is a beautiful specimen. If I can pat myself on the back, I love how it came out, but I cannot get down to the species. And it's only because it was to key it out at the species level, I needed the flower, but we did not have it blooming. And even though it's amazing, its application is more limited because of that. However, I don't want to discourage you from taking photos of even dead things because any information is good information. But the best information will be with flowers, in my opinion. I could be wrong, but we definitely have had to iNat dead things now that things are scorching. And we've had lovely people in the iNat community go through and have fun identifying all the dead things, because you still have a lot of those morphological traits, you know, like not flower shape, some dead fruits possibly, or even the shape of the leaves. But if you were going to get really close with it and really, really try and work for it, the flowers. Anybody else?
I always think, you know, something I feel like we all eventually did was a picture of the habit, like kind of a faraway picture that has some sort of size, picture that shows flowers, fruit, the underside of the flower, just any very specific characteristics, but far away pictures as well. Not too far away, of course, but just to give the habit, I think also very helpful. Yeah, shout out Sam biology.
Thank y'all very much. That's some great advice. I'm going to keep going through here, and I've saved the chat. So there are some wonderful, there's some accolades in here. Some folks are saying thank you for your presentation and saying you've done a wonderful job, and it's fantastic to see young scientists interested in botany. And thanking you for your work here with us at the garden. And Britt. Lisa asks, what makes the coneflower you found differ from the more common ones?
We actually don't know. That's an Aaron question. Okay, yeah. So we actually didn't deal much with that coneflower. Whenever we saw it, I remember Brooke and Tiana were kind of discussing it, kind of looking at it from afar, but it was no longer in bloom. To me, they look the exact same from far away. I definitely don't have the expertise in that. And we had I believe she's a research associate. Her name's Erin, and she came out with us to look at them to identify the rare ones. So I can't tell you the exact difference, but yeah, sorry, that was a very long-winded way of saying we don't know.
That was an excellent question, and that's part of the problem with this rare species. It can be confused if it's not gotten in the right season. So some of the morphological characters were some of the hairs, and some of the other vegetative characters, which is what was available to us at the time. But you should hopefully see a presentation about that sometime in the future. So just keep following some of our reports out here. Let's see. Jean, you had a great question. You asked where exactly this park is in Tarrant County. And thank you, Rachel. Rachel shared an article describing where this location is. We're talking about, this is in Crowley, Texas. It's southeast of Benbrook Lake. There's more information in that article that Rachel posted, which I will post again in the chat for those of you that may have missed it. And then let's pop up in here. Lisa wants to note that bloom dates in her area are a couple weeks ahead of the DFW North Texas area. And Jeff Quayle would like to comment. Jeff is a botanist in the area, that there is a Native Prairies Association of Texas field trip there on the 21st of October that he will be leading. Jeff, if I can find that, if I can find a link to anything about that, I'll try to post it in the chat. Let's see. Lulu provides a little bit more information about getting photographs of the whole plant, the flower from the front and the back, the leaves, and a good shot of the stem. So I see y'all nodding, our interns nodding, and they very much agree with you, Lulu. Jeff is also making a comment about the rare species Echinacea rubens, which has pressed hairs on the stem, distinguishing it from some of the other easily mistaken species. And then Lulu would also like us to say that you don't have to know everything, but the willingness to learn is everything. So we still have some time for questions. If anybody would like to ask a question, if you aren't able to type it into the chat, if you would like to unmute yourself, then I think we have just given you permissions to do so. So please go ahead and unmute yourself if you would like to ask a question. All right, Katie, that's an excellent question. All right, I think we'll have to have each of you answer it one by one, but Katie's question is, what are each of you doing next? So if you'd like to come up one by one and talk.
So next, I'm interpreting that as you mean like after the internship. After the, is that how y'all took it too? Okay, cool. So after this internship, I'm going to keep applying for jobs. I'm hoping to either get into some collections work or, working with just natural history. I'm a huge natural history buff. So wherever somewhere will take me, I'm excited to just grow and learn since I'm a recent college graduate. The world is my oyster, and I just need opportunities like this to continue to learn and further specialize in what I love and to learn more things. So yeah, this internship has given me a huge boon when it comes to being able to do field work and more research experience, which is always needed. So that's me in particular. So
So I still have about a year and a half left till I graduate. I'm majoring in bioenvironmental science with a minor in ecology and conservation biology. In terms of long-term career, I'm still trying to figure that out, but after this internship, I'm headed back to A&M and I'm actually currently working there part-time at the SM Tracy Herbarium as a digitization and imaging assistant.
I have an additional semester to finish at college, and then ideally I would want to, starting after discussing with them and people that we were so lucky to meet and talk to throughout Brit and beyond, I want to continue my education, maybe get my master's, and ideally looking for something towards the archival industry, museum type thing. So we've had a networking tea with someone who went through and got their master's and doctorate in library information system or science. Information science, library information science. So maybe that, but other than that, who knows?
So I actually just accepted a position with AmeriCorps. I'm going to be the DFW Conservation Fellow, kind of like working on increasing green spaces in the Metroplex. And that's about a year-long position. I'm also excited because I'm going to be a part of the fall Master Naturalist course. So I'll be doing that. And then ideally, in like after maybe my position at AmeriCorps, I would love to pursue a master's. I'm not really sure in what, but I'm really interested in like ecological restoration and conservation. So yeah.
Thank y'all very much. It is exciting. This has been a 10-week internship, and y'all have seen how our interns have bonded with one another, and we're gonna miss them very much. So there's another question in here. Lisa would like to know, are you considering joining us on Armchair Botanist in the future? And will you be spreading the word about Brit, Herbaria, iNaturalist, and Bio Blitzes to your classmates?
Yeah, no, I think we can all say definitely yes. Yeah, all of the ways that we like learned to interact with collections and conservation, I think were brilliant, and it was my first time ever interacting with most of those.
Oh, same.
So
and we definitely all loved our time at the Brit and would love to continue collaborating or volunteering here. So
completely.
for sure.
Absolutely,
Yeah.
especially since like I know a couple of us are local.
Yeah.
I know I definitely keep telling I went to go tell my family who they're more into like birds and stuff like that. But I told my dad, I was like, you really need to get on iNaturalist. And turns out he's been on iNaturalist for years now. So whenever I can get on iNaturalist, like this is the first time I ever heard about iNaturalist. I think I used it for like really pretty photos at one point, but this is the first time I've actually used it. And my dad has just peppered our backyard in observation points. And I've definitely told my family about, post-retirement coming here to volunteer with like the mounting staff because it was so much fun. And whenever we got to do it, or the imaging, I loved doing that whenever we had the opportunity. Yeah. So yeah, and even Armchair Botany, that'd be a great opportunity.
This is my third summer at Brit, and at the end of every summer, I'm like, oh man, how can I find a way to come back? So I'm definitely a big advocate of Brit. Tell people to come volunteer whenever they can.
Thank you. And on that note, Kate, Linda Jo is one of our regular Armchair Botanists and is on the Zoom session today and is really excited to see you at Tracy, back at the Tracy Herbarium. I'm not sure if the two of y'all have interacted yet, but she's looking forward to seeing you.
Awesome. I'll see you there. I'll definitely ask my boss about getting together. Yeah.
That'd be great.
And last in here, Lisa also wants everyone to know that Texas Parks and Wildlife Department uses iNat data for their conservation plan submitted for state and federal government for conservation funding. So it is a very important tool that everyone can contribute to. Are there any other questions that anybody would like to ask on the call? You can unmute yourself if you wish, or you can submit it in the chat and I'll ask it out loud for you. There are a lot more well wishes coming in next. All right, well, thank you ladies very much for your presentation. I will stay on the line a little bit longer because we may have some questions from our armchair botanists that are transcribing specimens. So everyone is welcome to stay. But best wishes for everyone. Sue Prairie would like to know what's next for transcription. This is a big thank you to, I hope all of you that are armchair botanists on this session were able to see the impact you're having. The work you have done made a lot of the interns' work this summer possible for looking for specimens online in Torch. You heard that, I think, from everyone today. And Sue's question is, what's next for transcription? We have a currently funded National Science Foundation grant to digitize specimens in Texas and Oklahoma. This is the project that Kate is working on that maybe Gabby will get involved in when she goes back up to Oklahoma State, because they are also partners. So we will continue to, we have a bit more transcription to complete, which is pulling data off labels into a database. The next step will be to georeference. So the idea behind that is that many of the collections that we are dealing with were collected without capturing any geo coordinates. And so that's going to be the next step. We're not quite ready to release that full throttle to armchair botanists, but it's the first place we'll go when we are. So stay posted for the georeferencing edition of Armchair Botany coming out before the end of the year. Hopefully that answered your question, Sue. Any other questions, everybody?
I just unmuted myself. This is Linda Jo. And I have noticed some of the specimens lately have got the chromosome or whatever. I'm not familiar with all that stuff, but should that be transcribed, and where should it be put?
That is an excellent question, Linda Jo. So some of those chromosome counts may appear as a separate annotation label, in which case you can mark that specimen as expert required. I believe that's what Ashley's having you do when there's some of that extra information. So if you do that for now, Ashley will respond to you. But there is one of the fields into which you can put those data, but just mark it expert required for right now. And Katie, I don't know if you're still on this chat, but the primary way we are transcribing specimens is through the Torch portal, which we've also been calling Symbiota, which is where Kate has been working this summer in the database. And so that's what Linda Jo's referring to. Thank you, Linda Jo. Hopefully that answered that.
Thanks.
Are there any other questions from our armchair botanist or from anyone for our interns or about transcribing?
No, but are you going to be in McAllen in October?
That's a wonderful question, Linda Jo. So I'm sure there were many of you in the audience that were cheering Jade on when she said she was going to be in the Master Naturalist program this fall. The Master Naturalist program is just an incredible program. And what Linda Jo is referring to in McAllen is the annual meeting. And I'm sorry for those of you that missed it because registration has closed, I believe. But no, I unfortunately am not Linda Jo. However, we have two wonderful representatives from our program, Ashley Bordelon, of course, who you know, who are missing today, who's your regular host, and Kimberly Sasson, who's perhaps still on this chat. So we will be represented, and you will have two workshops from the Garden and Brit.
Yes, I've signed up for them.
Oh, we're excited. I'm sorry I'll be missing you. It's just
I'll miss you.
Thank you. All right, if there are no questions, I'd like to thank all of you for attending our session today. Interns, is there anything else you'd like to say? Are you good? All right.
Just thank you for the opportunity to present. I know we all we all need the practice.
Yeah,
And
it was really fun.
we're just so happy that we can present the work that we've been doing all summer. It was amazing to see it come together. Never worked with a better group of girls or just anybody on a collaborative project. It's been amazing, and we really thank everybody for allowing us to do this.
And for coming and watching this.
Yeah, facts, coming and watching us and being there to support us, and we're incredibly thankful.
Tiana.
Yes, Lisa, did you want to ask something?
Yeah, I was just going to tell him that Oklahoma also has a Master Naturalist program, as the many other states. So if you're not, you're welcome to any of them. And if you're not in Texas, there are other outlets that are similar.
Yeah, good to know.
Thank you, Lisa. That's wonderful information and a good reminder. All right, well, it is 12:56, so thank you to everyone for attending, and stay tuned. Please go to that URL I shared earlier. If you just Google Armchair Botanist on the Fort Worth Botanic Garden webpage, you will find our page, which will list the events that are coming up. So once a month, it's the Armchair Botanist Forum, and hopefully we'll have some of our interns come back and present in the future about work they're doing. So we would love to have that. Thank you very much, everybody, and bye-bye.
Bye, thank you.